in class, and our projector is not working, so it's pointless for me to, I guess I'll record. We'll talk through this, but I guess I'll be able to record the slides so you could review this, but we just can't see them. And I'll try to help guide us through that. I'm sorry. So here we are back, and we have, first thing to say is we just have a slide that says register for summer classes if you're interested in taking summer classes, because some of the courses have low enrollments and those courses will get canceled in the next couple of weeks if students don't register. So if you're thinking about taking for summer classes, take them. Okay, what should you do this week? So we're covering chapter 19, and you're working on your term papers, and some students already finished theirs, and but you're plugging away at those because those are due at the end of next week. And if you're in the 4-H STEM Ambassador program, you're just continuing to work on that to Sarah Sparks' approval. And you know, if you just do your responsibilities, Sarah will say you did a great job. So you got to complete that if you're doing that, or write a term paper, one or the other. Then our test is going to cover chapters 18, 19, and 20. So I want to talk a little bit about chapter 20. Chapter 20 is about cancer. It's really divided into two parts. The first part describes oncogenes and retroviruses and retroviral oncogenes and signaling pathways and things like, it introduces a couple of new proteins like June and FOS, but it goes back to things like RAS and ERB and so forth. And so I know that, you know, that's a whole, you know, another big chapter that, you know, we have before our exam. And huge chunks of it should be familiar like that and adding to them, but also pulling things together for you, like a somewhat of a review. And so that part, especially, there's some nice add-ins to it. It adds some more stories, a couple more stories like that about oncogenes and their role in cancer biology like that. The second part is about cancer therapeutics. And that adds a couple of lists of different therapeutics, but we have our go-tos, and our go-tos are tamoxifen. So tamoxifen is one that we go into that part of that chapter understanding. It only works on some cancers, and that's true for all of these molecular therapeutics. It works on some cancers, the cancers that have mutations in the pathways that that therapeutic affects. Then CAR T-cell therapy is covered in that. Then cetuximab is described in that. And cetuximab is the monoclonal antibody that will stick to ERB that's mutated and block its uncontrolled activity. And it can only be used, you can only use cetuximab if the downstream RAS is wild type, is not mutated. So the logic of that second part, we have examples of. And we're not going to try to stack a whole bunch of more examples on them, but in reading through all that chapter, you should be saying, okay, I have framework for this, and I'll add a little bit of information, but I'll especially understand stuff that we've been talking about really well. So I think chapter 20, I definitely encourage you, you know, if you've gotten used to saying, I'm just going to rely on the class and I'm kind of glance at the book, read chapter 20, because it is a really good summation of what we've been trying to do, you know, all from day one. Yeah, Hannah has a question?
So for this coming, because we have tumor, we have like the exam for and then like the final exam. Does the final exam have chapter 20 or is it also?
Yep, the final exam covers everything. So that's a reason to study a lot for this exam, is because we're going to have like 2 exams on this material.
But like exam 3 that's coming up in a little bit, that's going to have chapter 20 on it.
The final exam? Yes. Exam 2, a week from
Or
Thursday.
Sorry,
Yes, it's going to be 18, 19, 20. That's our new chapters, like that. And I won't explicitly ask stuff from the first part of our course, but it's just built up, right? So some students will think they should reread. Well, actually, you might want to review the muscle videos from the first part of our course, because chapter 19 talks about them. Talks about muscle differentiation and satellite stem cells. So I won't be taking any stuff, that's back for the first exam, for this exam on next Thursday. It'll be that new material, but some of that new material requires you to really understand the first part of our course, and so as I've emphasized, make that review video for the first exam to understand that material. You'll need to understand that material to really launch into the new material and do well on a second exam. Then I try hard to write a second exam that you can finish in time. It'll be 50 questions, and they'll be hard questions like last time, but I'll try to write it so so some of the answers there are shorter, and there are just a few answers that would be a little bit longer. Keep in mind, what we said all along, that there are always kind of a direct, short answer to all the material in our course, but then we do longer explanations of them, like that. And so lots of students did that on the first exam, where they went into pretty long explanations for what is really a targeted thing. And that's fine, but just you have to have that kind of go for it mentality. If you understand it, be confident, and just go boom, 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 there you go. And so let me just say, some of this involves recasting or rewriting, rethinking what we've learned about in class. And I gave some examples of that, where I say, well, what are the targets for the CDKs at start? What are the targets for the CDKs at the G2 to M transition? And it's just boom, 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 like that. Sometimes no explanation. I'll say explanation, and if there's an explanation, you need that, or drawing, or stuff like that. Like that? Yeah.
Between the second exam and the final, are we covering any new information?
Yeah, there'll be like one new chapter, and it'll probably be we'll talk about it, but it'll probably be this chapter on the nucleus that's back in the third section, I think is chapter 12. Maybe one chapter, maybe 1.5 chapters, but yep, there'll be new material. But then our final exam will be a real just straight questions, like the kind of questions that we have in the second exam, not broader questions, not summary questions, but just like ding, 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 ding for the final. And it'll be then sweeping back all the way from day one, all the way cumulative final. And it'll have some new material, but not very much. It'll just have lots of stuff from throughout our course. Yep. Yeah. So again, our exam's going to cover 18, 19, 20. We're done talking about 18, still answer questions. Then we're talking about 19 this week and getting into chapter 20 as well. Okay? All right. So other questions people have about anything at all? The next thing is we're going to talk, and you'll see, we just breezed through programmed cell death. We already have established a kind of nice framework for it. There's autophagy and there's apoptosis. And we knew about 2 points of information about autophagy, right? 1 or 2 questions on the test about autophagy. Just what I talked about in class, because you're not required to read about it in our book. It's dealt with in a different chapter. But it's important to compare and contrast it. And then everything we talk about is apoptosis. And where we got so far is this idea that for apoptosis, another cell is going to phagocytize the dying cell. The cell's dying, and it's choosing to activate programmed cell death pathways in itself, and part of that includes signaling another cell to eat it and recycle the components. So that's the big distinction between apoptosis and autophagy. And then at the end of class, we learned how a cell signals to get eaten, and that is it flips out phosphatidylserine phospholipids in its cell membrane, which are inside on the inner leaflet of every one of our cells and every animal cell on our planet right now. Every normal, healthy cell has a variety of phospholipids that make up the cell membrane, and the inner leaflet, the inner layer, some of those phospholipids are phosphatidylserine. And any animal can flip those out. They don't flip out spontaneously because the phospholipid head would prevent that from ever happening, almost ever happening. And instead, they activate a protein. And the protein is called scramblase. And so, for apoptosis, that's a key hallmark feature of that, is apoptosis involves activating this scramblase, and this scramblase is a protein that grabs phosphatidylserine and helps flip it out. And you can just imagine what it does is it covers up the hydrophilic head and pulls it through the membrane, and so that now the phosphatidylserine is on the outside. And that's a hard phosphatidylserine is a hard thing to learn, but you'll learn that for our exam because it's a really good example. You're used to thinking about phospholipids as playing a structural role in membranes, they do. Then you're used to thinking about lipids as playing a metabolic role, right? Fats and lipids, like that. Lipids also play information roles. Here's a lipid, a phospholipid, in a normal cell membrane, playing an informational role, like that. So really interesting, right? And really significant. There are lots of other lipids that do that, like estrogen and testosterone are lipids that are hormones, right? So this is just a little taste of that, so it's important for a variety of different reasons. You know, really scramble to pick and choose highlights to introduce really important concepts, and that's one. Lipids playing a role in structure, metabolism, and information flow. So the scramblase gets activated, and who activates it is a protein called caspase.
Can you repeat that again? You said lipids play a role in structure.
Structure, metabolism, and information. Does that make sense, right? You're used to that structure. Oh, membrane, you know, actual physical role because of their chemistry. Then metabolism, of course, you know, they have a lot of caloric energy in them when we metabolize them. And then here's this one, information, which is just a striking example. And I said, every cell in our body, all the time, every animal cell. Did T-Rex have phosphatidylserine? No doubt. Because every vertebrate has it, right? Like that. And even invertebrates have it, right? So everything alive, all the animals alive on our planet are using this as a plan to identify cells that are going through apoptosis and destroying them and recycling them. Okay, so caspases do that. Then caspases are a family. So they're paralogs, and they're named by just numbering them. In humans, there's 1 through 9. We don't have to worry about the numbers, but we do need to understand it's a family. It's a family of proteins, and they're always present. They're always present. And these are what a person, and again, it's not a very commonly used term, but this is what people would call an effector. So we have a lot of regulators, and then there are some, what are they regulating? They're regulating effectors. Because when caspases are active, the cell is going to die. Caspase is like, sorry for the analogy, but it's the gun. And this is going to activate. So next thing is, what do caspases do? Caspases are enzymes, and they're enzymes that catalyze a reaction. And the reaction they catalyze is they catalyze cutting proteins. So they're called proteases. And proteases have as their target proteins. Some proteases just chew up the whole protein. They degrade the protein. Beta-catenin normally just gets degraded. MYC protein, very unstable, just gets degraded. And so when we think of proteases, we have to start out by thinking of these general proteases that just chew proteins up. We're going to get rid of this protein, we're going to chew it up with a protease. There are some proteases that have specific targets that just click. And that happens here, where in our cells right now, the caspase is inactive, and the scramblase has just an extra piece of protein. So this is an inactive scramblase, and it has an extra part of its protein that's just covering it up, covering up its binding site to the phosphatidylserine.
And that's the caspase.
And so this is the caspase, and the caspase's function is when it gets activated, it just clips that off, that specific part. And that allows scramblase to be activated. So it's an important concept because it's a different way of regulating a protein, where the intact protein is called a pre-protein. It's made and it's just got an extra piece on it, part of the protein, that's blocking its active activity. And it needs a specific protease to clip it. And that's what these caspases do, and they have several targets. Like that, yep.
So they call it a free protein, a piece that they cut off?
The whole protein is called a pre-protein,
Okay.
because it's the inactive pre-protein. So it's originally synthesized, it's present in every one of our cells right now. Our bodies are making it in every cell right now. They make it as this longer inactive pre-protein that is blocking its own activity. And all the caspase does is it gets activated and it just fights off that little extra piece. And these caspases have several different targets that lead to programmed cell death. And for our course, the book lists a bunch of them. And for our course, we will learn about 3 of the targets. So one of them is the scramblase that scrambles phosphatidylserine to the outside of the membrane. A second one is there are enzymes called, you can write them like this, DNase, because it's an enzyme that degrades DNA. So its longer name is a deoxyribonuclease. And what these do is they are enzymes that bite the DNA and they cut the DNA. Okay? These DNases, again, there are a variety of them, and they're used in the cell in a variety of ways. These are also made as an inactive pre-protein. They're in our cells right now. They're an inactive pre-protein waiting every day of our lives to be clipped by a caspase and become active. And when they're active, they just chew up all the DNA. And that's a good one for us to learn because this cell is killing itself. What is one way a cell could kill itself? And that is just destroy all the DNA, right? The cell needs the DNA to make the proteins and to make the other genes, and it's just wiping it out like that. Okay, so that's a good one to learn about. Another reason we learn about it, the book talks about it, is that that's easy to detect in an experiment. So you say, this cell might be dying. Whenever it's dying, you say, let's check its DNA, see if its DNA is intact. When the chromosomes are intact, they're very long, and if you run them on a gel electrophoresis, they only move slightly because they're so long. But when they start getting cut, they just get cut, and they start running down the gel faster. And where the DNases tend to cut them is in between the nucleosomes. So you get this ladder, like not just a smear of DNA running on a gel, but like a ladder-like pattern that's very distinct and indicative of apoptosis. So a couple of reasons why we'd want to learn that. And then the third one is there's a role for the mitochondria. And that's another good one for us to learn because you want mitochondria in a cell to live. And if you're going to die, just destroy the mitochondrial function. We'll see there are really 2 parts to this. One is that, understand you already have a cell needs mitochondria to make energy and make a lot of ATP, right? So if we start messing with the mitochondria, we're in trouble. The second one is the mitochondria are actually going to release some factors that add to the death process. So we can think of the apoptosis as, we're going to damage the mitochondria, and the mitochondria have been like a bag with dangerous stuff, and they're going to release the dangerous stuff. And so we'll talk about that in a little bit more detail. But those will be the 3. Caspases target these. So in each case, caspases target a protein, and we don't need to learn the proteins about that one, just the idea that the mitochondria plays a role. The caspases are kind of downstream of this, so I should maybe not draw this line quite directly, because the caspases actually work on stuff that's coming out of the mitochondria, like that. Yeah, but that's a detail. Yep, let's see, there was a question.
Are caspases only active in apoptosis pathways, or do they have other functions?
It's always dangerous to say only, like that.
Sure.
But at this level for sure. When we think caspases, we think they're present right now, inactive, and if they get activated, it's because of apoptosis. One thing we can do is we can compare and contrast autophagy and apoptosis. Caspases have no role in autophagy. And that would be a question we could have on the test, and it would be like, give 3 differences between autophagy and apoptosis, and you would probably just list things that are in apoptosis. Activate caspase, flip phosphatidylserine, cut up the DNA. Those happen in apoptosis, not in autophagy.
Are these autophagy?
These are apoptosis. Yep, almost everything we'll learn is apoptosis. Autophagy we talked about last time just briefly, and that's where the cell eats itself inside. So it creates this autophagosome, which is like a little internal Pac-Man that just starts grabbing and engulfing organelles and ribosomes and things like that, and just recycling them. And it can do that a little bit under conditions of malnourishment, or it can do it a lot under conditions of, say, like insect metamorphosis, when the caterpillar is just going to get turned into liquid. That's a lot of autophagy. Yeah, Hannah?
How do castases get activated? Do they have
Yes.
another caspase cutting the caspase?
That's a good question, and the answer is we get really interested in that, right? This is very different than the chapter 18 story about the CDKs. But remember, the CDKs were effectors. We talked about target, 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 and you're just like, I see how that's working. These are the real effectors. There's a lot of regulation upstream of them, because they're there in our cells right now, and they need to be inhibited all the time. And so the real story of apoptosis is upstream regulation. There are proteins inside our cells right now that are keeping the caspases inactive. And if we can either encourage the activation of them or the inhibition of them. Okay? So that's why we're starting here, is to see that. Okay? And so we'll talk a little bit more about these, but we're pretty set on the phosphatidylserine story, and already pretty set on the DNase one. Those are just dramatic examples where you're like, oh my gosh, I see what's happening there. We'll loop back to the mitochondria when we add a little bit. And there's a nice figure in your book that shows a mitochondria, right? It's a real cell biology figure. There's a mitochondria, and here's some factors that come out of it and regulate caspase activation. There's some positive feedback loops in this, where caspases, like Hannah kind of said, caspases help activate more of the caspase activity and things like that, and that's happening some in the mitochondria. But really, the upstream regulation is some other proteins. Yeah?
I just wanted to make sure that it was proteases that were targets of caspases and not just scramblers.
The protease, the caspase is a protease.
Okay, so then is it targeting all proteins, or is it only targeting scramblers?
Specific proteins. That's a good question. So there are a variety of enzymes that destroy proteins. And like I described, some of them are just general. They just chew up proteins and recycle the amino acids. Like we've seen that in the Wnt signaling pathway, this transcription factor beta-catenin is unstable. And it's just getting chewed up and recycled like that. There are other proteases that have very specific targets, and that's what the caspases are, is they have a set of very specific targets, and those are inactive pre-proteins, and the caspase is a protease that just clips that part of the pre-protein that's keeping it inactive. So we just have 2 examples of that. There are several examples, and several other things that are happening. Our book talks about that the nuclear membrane gets destroyed. And it talks about that. That's a add-on like that. If we could get these core pieces out, that'd be great. Okay? So then the next thing to say is that of the upstream active regulators, there's one that is world famous, and it's called Bcl2. And it's an inhibitor of caspases, and it's always in all of our cells, and it's inhibiting. the caspases. Yep.
Is that Bcl2?
Bcl2. And so for this, our key players are that family of caspases and what we've learned about them already, and we'll learn a little bit more about them. And then the next thing is we want to know the name Bcl2. And Bcl2 is an inhibitor of apoptosis. And so apoptosis involves release from inhibition. The caspase effectors, the killers, are there and are being inhibited. So it's a little curious to see that, that the story is really a release of inhibition. You can think of Bcl2 like the safety on a gun, right? And you got to release the safety for the gun to shoot, like that, right? There's a lot of control, regulation of that. This inhibition can be released either internally, so within the cell, and we've seen that, there could be DNA damage that didn't get fixed, and so it's going to go through apoptosis, it's going to die. And that happens where the cell says, I'm out of line, I'm going to wipe myself out. Or there could be external signals. So there could be a signaling factor that works through a receptor that's targeting this Bcl2. So it's a pathway, and we can activate that pathway and apoptosis either through a problem inside the cell or through signals outside the cell. Okay? Then,
What's an example of an external influence?
like a hormone. Like in insect metamorphosis, when the caterpillar destroys itself, it's through hormone signaling to release the inhibition of apoptosis or autophagy too. Autophagies can be either internal or externally regulated like that. So signals like hormones and other factors like that can trigger apoptosis. Another example, just to bring it up, is that in the CAR T cell story, those CAR T cells are releasing factors. Those include factors that encourage other cells to kill, and they do that by sometimes phagocytizing, but other times releasing factors that say to the target cell, you start killing yourself and we're going to eat you in the process. So there are lots of examples of these external factors, either at the level of a hormone or more of a local paracrine signal thing. Okay? So here's the next concept, and it's a thing where it's a stumbling block, but we're just going to breeze through it. This Bcl2, it's a protein, so obviously there's a Bcl2 gene, and that's a member of a gene family. And I don't know, there may be 18 members in humans. It's a pretty big gene family. And they have stupid names, just a bunch of stupid, goofy names. And we don't have to learn any of them. Here's what we have to learn, is that in this gene family, these are all regulators of apoptosis, and some are anti-apoptotic, and some are pro-apoptotic. So that just makes it confusing. What people call this gene family, they call it the Bcl2 gene family, because that was the first member and the most famous member discovered. And then we just have to take it easy because some are pro-apoptotic and some are anti-apoptotic. And that's confusing, right? Because they're inhibitors, and some of them are inhibitors of inhibitors. like that, you know? So you're like, So we don't need to learn the names of the other ones, just that there's a gene family. We need to we need to remember again what a gene family is. It's a set of paralogs, two genes in the same species that share a common ancestral gene. The gene family arose by accidental gene duplication in evolution, and the existence of it is evidence that this is an important pathway to regulate. And as animals evolved, they used the Bcl2 regulation and evolved more and more complicated ways of regulating it. So in a person, there's a bunch of these in this gene family. In a real simple invertebrate, like a worm, there's usually only one, and it's Bcl2. And they just don't have a sophisticated regulation of apoptosis in their simple bodies that live, short lives with relative simplicity, like that. So that's that Bcl2 gene family. Let me just find a slide just to read you some of the names.
I have a question.
Yep.
The anti-apoptotic, are those the ones that inhibit the inhibitors?
Yes.
Okay.
Yeah, and our book has, here's some pro-apoptotic names. BID, BAD, Noxa, Puma, BIM. And you're like, okay, not interested. And the book has figures, tables of the family, and maps of the proteins, right? Because it's a very important part of human health. But we don't have to get into the weeds, just the understanding of that there is this family that's regulating the caspases all the time. And Bcl2 plays a really important inhibitory role. And the next thing to say about it is that there, again, are orthologs of Bcl2 and caspases in every animal. So even in a simple worm, there are those members of this story, the caspase activators and the Bcl2 inhibitors. And the release of apoptosis is to remove that inhibition. Like that, okay? And our book has some nice pictures of that and stuff like that. The book wants to talk about the people who studied a simple worm at MIT in the 1990s and discovered these orthologs in a simple model organism. So there's a page in a chapter about this scientist at MIT named Bob Horvitz, and he and others won the Nobel Prize back in about 2000 for this work. So part of the chapter also is underscoring that model organism approach, like that, and the book wants to talk about that. And we want to read about that because it's significant, and what we just want to underscore again is the understanding of ortholog. So in a human and that little nematode worm, there are orthologs of Bcl2, two genes in different species that are understood to have a common ancestral gene. They understand a common ancestral gene for two reasons. One, the proteins are really doing the same thing, inhibiting caspases in the little nematode and in the human. And two, if you look at their amino acid sequence, they're so remarkably similar, you couldn't expect that to have ever occurred in the entire universe by chance. Right? So we're back to that concept that we learned about in the first exam, but it comes up here and underscored again and again, because that's one of the things that you want to take to any party you ever go to and say, oh, you want to talk about evolution? We just talk about that for the rest of this party, like that. So that's coming up, right? Then again, caspases are a gene family, right? In some simple organism, there might be one caspase. In humans, there are 9. Bcl2, maybe 1 in simple organisms, maybe 15 in us, like that, okay? And paralogs are genes within the same species that are understood to have gone through a random gene duplication, but otherwise share a common ancestral gene back in the day, million years ago. Yeah, so the distinction, just say the definition. Orthologs are two genes in different species. So you have to name the species. And they're understood to have shared a common ancestral gene in the last common ancestor of those two species. Paralogs are two genes within the same species. and they're understood to have had a common ancestral gene that duplicated. And it was a natural selection advantage to have two of those and have them become slightly different. The way to think about it is like a set of screwdrivers, and some people have a lot of screwdrivers, right? Those are paralogs. They're all screwdrivers. They're doing the same kind of thing, but they have slightly different structures to play slightly different roles. Okay? All right, so let us just talk about this Bcl2. So Bcl2 is an anti-apoptotic protein. And there's some cancers that are called lymphomas. That's a kind of blood cancer. And there's some lymphomas that have a chromosomal translocation. And we don't have to remember the chromosomal translocations we've learned about for the second exam, but we'll have to remember them for the final. One chromosomal translocation we learned about was MYC. MYC is a transcription factor, so it's got a gene, it's a protein got a gene, and MYC is really unstable. But in some cases, in some leukemias, there's been a chromosomal translocation where there's the regulatory region of MYC that got replaced with a regulatory region for the antibody gene that's made at a high level in some blood cells. And now MYC is overexpressed. It's wild type MYC, normal MYC, but it's just way too much of it. That's one example. Then another chromosomal translocation we learned about was the woman who has skin cancer, and the chromosomal translocation, just an accidental mutation, repaired two broken chromosomes and brought together the regulatory region for skin collagen with the transcription unit for PDGF. And that woman is accidentally making lots of PDGF in the skin cells, right? So here comes another chromosomal translocation, and it's with the Bcl2 transcription unit. And this time it's a different cancer, but again, the regulatory region for an antibody gene. Antibody genes are made in blood cells, so we're going to have a lot of Bcl2 made now. In these specific kind of lymphomas, we have an overexpression of Bcl2.
So the cells aren't dying when they should be.
Yeah. Yep, it's normal Bcl2, you have too much of it, so they are too unwilling to die. They pick up other mutations, start to grow out of control, and they just don't die. People who have this lymphoma actually have a sort of an early phase in the disease where they can be spotted, where there's a little bit of out of control growth, could be going on for years, and they're concerned about it. And they already have this mutation, so there's just a little bit too much of this growth, and this is already mutated. These cells in a normal person, they should just be dying. They're not dying, and it's sort of a precursor to this period where they might get another mutation and the cells start to grow really fast, and they do not die, like that. Yep. And we'll put a specific name on it like that, but this is the thing, right? And it's just like, okay, I see how programmed cell death plays a role in some cancers. And here's just a really, really good example of it. Scientists, because it's a chromosomal translocation, you can see that under the microscope. During mitosis, when the cells are dividing, the chromosomes are condensed, and you can see the chromosomes, and you can see there's a specific problem with two chromosomes. And one of the chromosomes has the antibody gene, and the other has Bcl2, and they actually join together. So for years and years and years, doctors and scientists have diagnosed this cancer as having that chromosomal translocation. for years and years and years. That's why, that's it, it's either part of the diagnosis is you have this kind of lymphoma. We know it, we categorize it because we saw this chromosomal translocation. Then it was only in the last, 20 years they were able to analyze, well, what are the genes involved in this chromosomal translocation? And you see them and you're like, darn it, right? A gene, regulatory region, that is normally expressed in the blood cells at high levels. Antibodies are the specialty protein for those blood cells called B cells, white blood cells called B cells. That's their job. Make a ton of this. And we've accidentally joined that to the Bcl2 protein, a gene. And the Bcl2 was getting made in those cells, but now it's just getting made really high levels. And anti-apoptotic. Even if the pro-apoptotic factors like PUMA, BIM, BAD, those guys get activated, too much Bcl2, they can't overcome it like that. It just overwhelms the natural regulatory control. And so that's just a really satisfying example to fit into this normal regulatory pathway, to show how this pathway could get disrupted by mutations. We would consider Bcl2 to be like a sort of a tumor suppressor or play a role. It's not causing the cells to go out of control like that, but people don't want to call it a tumor suppressor because it's a different kind of role. So we have those terms, oncogenes like RAS that are in normal control pathways and if get mutated could lead to out of control. Then we have tumor suppressors like retinoblastoma that's always putting on the brakes like that. And then there are examples like this where you're like, it seems like a tumor suppressor, but I don't know, I wouldn't call it a tumor suppressor like that. So that's the kind of role that these programmed cell death pathways play in cancer, and that's why the topic is brought up in this fourth section of our book. Right? We got chapter 18, regulation of the cell cycle, and oncogenes causing out of control cell cycle growth, and then right after it, chapter 19, because there are a variety of examples in cancers where there's a problem with programmed cell death. The cell's growing out of control, and it should be able to identify that and just put the brakes on. Okay? And that's that. Then the next thing we want to talk about is that we learned in our chapter 18 about p53, and p53 is a transcription factor that gets activated. So let's just put this down here. We want to loop back to p53 just to add a little bit to it. So this is this p53 protein, and what we learned is that this protein plays a function at start, and when we usually think about start, we think about the red light turning green. And p53 plays a role kind of like that school patrol who comes out in the street and says, nope, I don't care if the light is green, you're stopping. p53 gets activated at start by DNA damage, and p53 is a transcription factor. And when it's activated, it binds to a target gene. And we know all about this kind of pathway in general. It must bind to the enhancer, and it binds to the enhancer of the P21 target gene. It has other target genes, but this is a really nice one for us to learn because P21 codes for a protein that is a small inhibitory protein. It is a school patrol flag that can bind to CDK, and even if cyclins are present and the phosphates are organized properly, this just blocks start. So we stop and we try to repair the DNA. The p53 gets activated by phosphorylation, and what we want to add to now is over time, the p53 gets more phosphates added, just more and more, so it gets hyperphosphorylated over time. And when that happens, it starts to bind to additional target genes. So here's this enhancer, like that, and it's only the hyperphosphorylated p53 binds to it. And these are genes that code for proteins that are pro-apoptotic members of the Bcl2 gene family. And that's what we call them, and we don't have to learn their names like Puma and Bid and Bad.
Is that what it means when a gene is upregulated?
Yes. These genes get upregulated, they get activated, get expressed. So this p53 plays this dual role, right? It's like this school patrol that comes out and says, you know, stop, wait, and they wait, and they wait, and they wait, and they just eventually just start taking the flag and just smashing your car with it and saying, you're not going anywhere, you're dead. You know, like that. Get you out of the car and just whack you with the pole. And you're just like, well, that seems extreme. How many kids are coming to school? They're like, too many! You know, Dave. Right? And so that figures in our book in this chapter, and it's just a really satisfying add-on. We talked about that before, that p53 activates this DNA damage checkpoint at start, and the cells are, you know, sitting at start, waiting for repair. And if it doesn't get repaired in a reasonable length of time, that cell goes through apoptosis by an internal signal. That's again called an internal signal, right? I've got DNA damage, it's not being repaired, I've got issues, I'm out of here. I'm going to activate the caspases, destroy my DNA, wreck my mitochondria, do lots of other troubling things, flip my phosphatidylserines out and be like, I just couldn't fix the problem, right? That's like your washing machine, right? The lid is up and it waits for a couple hours and then it just catches fire. Just these clothes, you're not going to wear these clothes again, right? 'Cause I'm tired of this. I'm tired of waiting around. Yeah. So we want to know that piece. It's just a little capsule piece in there. And, you know, we can point to it now and say again, the targets are pro-apoptotic factors. Those Bcl2 right there, it's a gene family. They code for a family of proteins, and these are pro-apoptotic, right? What if you had DNA damage in that cancerous cell that was overexpressing Bcl2? You just roll on. Oh, there's some mutations. I don't care. We're good, right? See, because there's too much Bcl2, so even when you express the pro-apoptotic factors, it's just like whatever. I think things are good here, you know? And it rolls on, and that's one way that cancers pick up more and more mutations, is they start to have problems repairing DNA damage, or the mitotic spindle, keeping track of their chromosomes, and they just get sloppy, and that just increases the likelihood that they'll make more mistakes. There'll be something completely different. Like they might come up with a mutation that leads them to become malignant and metastasize to other areas, right? They were hanging out, but they just picked up a bunch of other garbage mutations, and they're just like, you know, they're not trying to be bad. It's just a natural selection process and evolution, survival of the fittest kind of thing. And this would be an example of that. Yes?
So when we're talking about the lymphoma, are we talking specifically about anti-apoptotic Bcl2?
Yes.
Okay.
For that lymphoma, the thing we want to learn about it is that there's been a chromosomal translocation that leads to an overexpression of Bcl2 protein and too much inhibition of apoptosis. Okay? So that loops us back to chapter 18, and our book has nice figures about that in chapter 19. It shows that visually really well, and we just get back to that because we understand it a little bit more. And again, we don't need to name Puma, Bad, just these are pro-apoptotic factors, right? It's not Bcl2 we're activating family members that would be trying to kill the cell. Yep.
When DNA is damaged and it's at one of those checkpoints or the control point, is that when it's more likely to pick up a mutation when the cell's trying to fix it, or
Yeah,
does it happen
yeah, right. So mutations are always random errors, and Some of them occur during DNA replication, like that, or during the cell's life. There's some damage due to some UV light, and the cell will try to repair it, right? And so there were these repair mechanisms, and they're normally pretty good. And if they weren't good, a person would be more likely to have mutations, and if they're more likely to have random mutations, they're more likely to have a bad mutation that happens to cause real trouble, like that, right? And so that's part of the story of cancer progression, is this idea of things get worse, and they get worse because there are more mutations in other parts of the cell's biology. And if we have problems with DNA repair, that would increase the rate of mutation, and that it would increase the likelihood that they would get more trouble happening in the cancer cell. And so that's a big part of this story that people always talk about, that cancer is a progressive disease. Starts out, there's some problems, and they can get worse over time. It just gets worse, and then cancer spreads and so forth, like that, grows faster and stuff like that. Yep. Okay, so there's also a figure in the chapter that shows, one of these, and we looked at this briefly, but here's one of those regulatory pathways that's going to stimulate something like FOXO or something like RAS, RAF, MEK, ERK, ELK. And I said, really typically, those pathways also lead to regulation of pro-apoptotic responses or anti-apoptotic responses. And we'll look at that throughout our book. Very often, the author's been showing that, right? They've showed complicated pathways, and we've tried to cut it down, to the bare minimum. But the author will show, actually, this pathway, this receptor has 2 signal transduction pathways, and it includes both stimulating the cell cycle at start and also regulating these apoptotic factors, sometimes increasing the likelihood of apoptosis, saying, if you're going to start growing, you have to be careful and increase, your likelihood that you go through apoptosis. In other cases, they inhibit, and it just becomes complicated. But we want to have that awareness that when we're getting signaling factors regulating cellular responses, especially things like growth, they're also affecting this mixture of proteins that are upstream of caspase. And this mixture of proteins is a little bit like that Bcl2 gene family. Those protein products are a little bit kind of like a majority rules sort of thing. We're like, let's vote. Let's get some voting going here. Who thinks we should, activate caspases? Okay, Puma does. Cool. And then who thinks we shouldn't? Oh, Bcl2? No? Okay, all right. Well, let's keep track of that overall level of those activities, right? It just gets complicated, especially in humans, with our big, sophisticated, long-lived bodies, a lot of that interplay at that level. So we don't need to dive into that detail, just need to know it exists. Then I've got a slide about that mitochondria. I think I'll wait until Thursday to show that slide and just look at this piece here a little bit, like that. And what happens downstream of that, what gets released and stuff like that. And I wanted to get to that because that's really our programmed cell death story, right there. And that the next part of the chapter is about stem cells, like that. So on Thursday, right, you want to bring questions, and next Tuesday too, questions about that. But the programmed cell death story is a nice package. The book gets kind of derailed by the complexity of the Bcl2 gene family, but if you just put a box around that, keep it simple, then the stories are very nice, very physical, sensible stories that really build on the principles, right? P53 and that chromosomal translocation story, like that. Yep. Orthologs, paralogs, So that's programmed cell death stuff. Do people have questions about that? Yep.
Would you mind going over this very, very briefly again, this side of the board? Like
Yes, the
27
P53.
synthesis? Yes.
Yes. Yeah. So this is this thing that's really from chapter 18, where we were talking about the cell cycle. And in chapter 18, we said that the big story in the cell cycle is this start. And that's where we always say red light, green light. And the light is green when the CDKs are active. And when they're active, they're kinases that phosphorylate targets like MCM, P27, and retinoblastoma, like that. So a bunch of targets that actually make the cell go into S phase. And that's the start. And upstream of that, there's a lot of signal regulation, like so when we say, oh, look at this growth factor binding to this receptor tyrosine kinase and activating RAS, RAF, MEK, ERK, ELK. Well, that ELK transcription factor, what is it bound to? And the answer is cyclins, like that. So that the cyclins are necessary for the CDK1s to be activated. So we go through a lot of talk about that start. And then we say, it's really, there's a lot to it, right? Like that. So we say, well, let's go over here to the G2 to M transition, and we'll learn a nice little package about the CDK that's active over here, which is CDK1 detail. And that's an interesting one because there are no small inhibitory proteins involved. The cycline's already there. And the issue is really, we just got to remove 2 inhibitory phosphates from the CDK, and we'll roll into M phase, like that. And who removes them is this CDC25 phosphatase. And so that's our story. And if there's DNA damage, we should wait. Like the washing machine with the lid up. We should just wait till the damage gets corrected before we roll into M phase. And the way we're going to wait is we're going to have check 1 phosphorylate CDC25. And when that's occurring, it's inactive. And we're just waiting. So you could, list really easily, what's the status in a cell that's arrested at the G2 to M1 transition? And you would say, well, check 1 is active, CDC25 is phosphorylated, and there are inhibitory phosphates on the CDK. And then flip over to when it's not, when it's rolling forward, what's that? So that's a nice, crisp package. And this is complicated. Just the start, turning the red light to green is complicated stuff. And that just means lots of different information, like that. Within that, we have a DNA damage checkpoint. And that's this one. It's different. It's a different pathway. There's no CDC25 story like that. And instead of check 1, upstream of this, it's check 2, and there's DNA damage though. And P53 gets phosphorylated. It's a transcription factor that's active when phosphorylated. We could go back to that list, we have going with transcription factors and how they're regulated, and we've learned several that are regulated by phosphorylation, like the STATs and FOXO and so forth. This would go there. If phosphorylated, this transcription factor is active. So when damaged, it's active, and it binds to P21 and other target genes. And P21 gets made, it's a small inhibitory protein, and it's like that. School patrol, right? The light's green, start, green, good, go, like that, wants to go, but this is saying no, stop. And as long as there's damage, the P53 is phosphorylated, we are making P21 and preventing the cell cycle to go forward. The add-on today from chapter 19 is over time, as this DNA damage is not being corrected, just over time, this protein continues to just get more and more phosphates. That's uncommon. Most proteins just have a single phosphate or 2, and that's it. But this protein gets hyperphosphorylated, and the more it gets hyperphosphorylated, the more likely it is to bind to these additional target genes, including these pro-apoptotic Bcl2 gene regulators. So it's saying 2 things. Initially it's saying wait, and then wait, and wait. And then over time it's also starting to say, we've been waiting a long time, and I'm getting tired of waiting, and we're obviously not going forward. I'm not giving up. I'm going to ask, you just to never go forward, and we'll go through apoptosis. Yeah. So it's satisfying, right? It's a really crisp molecular switch. P53 has this famous nickname, the guardian of the genome. It's one of the top 10 studied proteins of all time. Because it has other interesting stories, right? We're just learning to the intro, so just here's a couple of really crisp, real, principle-based ideas of P53 and P53 regulation and activity, right? It's a transcription factor, yay, The original clay model videos could have been about P53 instead of muscle, right? We could have picked so many different examples of showing transcription activation instead of muscle. Remember, the muscle videos are a universal example of transcription factor regulation of gene expression. Ellie.
Do you mind going over the last part of this image on the board? So like,
Yep.
is
We could reenact it with a lot of help.
when P53 is like hyperphosphorylated, is that just going to cause like the creation of a lot of the P21 gene? Is that what
No,
we're saying?
this is a different target gene.
Okay.
So this is these other target genes. So this is an additional target gene. One way to do this would be to label this, and again, you don't need to learn this, but just say that's PUMA. Okay? So early on, back here, early on, here's the PUMA gene. There's no P53 bound to it early because it's just got this single phosphate and it doesn't bind until it gets hyperphosphorylated.
Okay.
Right? So it's a time thing. Early on, some genes are activated, and those are genes that say, hey, wait. Then later, additional genes get activated and they say, hey, go through apoptosis. Yeah, it's really nice, right? You could draw it, you could say it, you could share it at a party. Tattoos, again, tattoos. All over. P53 here and there, and you're just like, look at me go. Guardian of the Genome, dude!
And then is it
Right?
going
I never wear a shirt.
is
Right.
PUMA eventually going to help like break down the damaged DNA? Is that kind of the
What would happen here? Yeah, who knows? What would happen there? What?
What was the question?
What happens? So you say, oh, okay, P53 gets hyperphosphorylated and we start expressing these pro-apoptotic factors. What would happen next? What?
Apoptosis?
Yes, along that path, what's the next step?
Spaces?
You have to activate other genes.
You have to inhibit the inhibitors. And that's what those guys do, is we have the Bcl2 playing an inhibitory role, and we're going to inhibit that inhibitor.
And those cat spaces are going to turn.
And then, yeah, and then we no longer inhibit the Cas bases, and they start to proteolytically clip scramblase and DNase and other targets, and the cell is dying. So this is upstream regulation, and there's this kind of balance between the Bcl2 family members, pro-apoptotic, anti-apoptotic. There's always Bcl2 anti-apoptotic. We can overcome it by making some of the pro-apoptotic family members. There's more regulation than that. It's not like our book shows this because it's true. Some of the factors just bind right to Bcl2, like bad binds right to Bcl2, and it's like, you're not doing your job anymore. And it's like two siblings, right? And they're like, I want to go out. And you're like, you're not going out. You have to stay home and do the dishes. Like that, right? That kind of direct interaction. There's more, it just spreads out into the cell, more aspects of this regulation. There are other inhibitors and stuff like that. But this is a central story in apoptosis, is the interplay between the pro-apoptotic and anti-apoptotic family members of Bcl2. Yep.
So, yeah, we see that Bcl2 has the pro-apoptotic and the anti-apoptotic. So that Bcl2 that you drew on the board right there is an anti-apoptotic.
Yes, so the original member of the family is Bcl2, and Bcl2 is a gene that codes for a protein, and the Bcl2 protein is an anti-apoptotic protein inhibitor. Then there are other gene family members, and some of those are also anti-apoptotic, but others are pro-apoptotic.
So when we have an overphosphorylated P53 binding to those target genes of pro-apoptotic of like the gene family of
Mhm.
BC1, 12 or whatever, does that mean it's not really called BC12 anymore? It's actually
So, yeah,
it's
so let's
actually
just clarify that, right? So here's a gene family, and the names are things like BIM, BAD, PUMA, so forth, Bcl2. Bcl2 is a gene, and it's one member of a gene family. And all of those code for proteins. And some of them play roles in inhibiting apoptosis, anti-apoptotic, some stimulating apoptosis, pro-apoptotic. I suppose the curious thing, but it's not that surprising, is that because Bcl2 was the original member, people just refer to the entire set of them as the Bcl2 gene family. Like that, right? Like I'm Dave, and I have a brother Craig and Barb, and we're in a family. You could say that's Dave's family because Dave's super cool. And, you know, kind of the original one you met. That's Dave's family, same thing. Just using Bcl2 in two different settings that you're trying to clarify, right? It's used both to describe this specific family member and then also the whole family member. And the way it's done is by saying Bcl2 or Bcl2 family, right? So like on the exam, you have to make, that's a really important distinction, right? Bcl2 is referring to this one, and it's anti-apoptotic. Bcl2 family is referring to all of them, some of which are anti and some are pro.
So like that would actually be PUMA, for example, up there, like activating the caspase.
So when some pro-apoptotic family members get made, they just bind right to Bcl2, just directly bind and inhibit it. And our book has a figure showing that.
And is that what PUMA is? It's like one of those inhibitors that binds?
Some are, some aren't.
Okay.
BAD is one I know that is bound directly, but I don't know the details of all of them. Yep. Yeah, well, I just see if it even says on here, because it has a figure of one binding directly, and we'll just see which one. Just hold on, come on. No, it's not naming them. I don't know if the book ever names them. In the figure, it's just showing pro-apoptotic binding to an anti-apoptotic family member. Yep. We just have a couple minutes left, so we don't have time for a concept chat today. But let's just check in to see if other people have any questions right now. So we have a couple more minutes. The next part of chapter 19 is also something where you put in a package because it's about stem cells, and we know a lot about one stem cell, and it's the satellite stem cells that are lining muscles. So those are kind of stem cells, and let's just get to this today. We'll talk about this, that in our bodies right now, in specific locations, we have some stem cells that are referred to as adult stem cells. And they're not limited to adults. They're present in a fetus, child, teen, adult. And our favorite example is the satellite stem cells that line all our muscles. There are others. We have them in our bone marrow, like that. Oh, sorry. Yeah. Right in. Yeah. Doing great. Everything's working out great. It's going out on the recording of the web. They're like, thank you. I missed class. Thanks so much. Yes, there was a question.
I was just gonna bring your
Bring that. Wake up, loser. And and we'll just see, distinct from them are embryonic stem cells that are present only in the very earliest embryo, just the first few days of pregnancy, and it's actually all the cells in the very first embryo, and it's only mammals. So that's a different kind of stem cell. We have adult stem cells, satellite stem cells, and then embryonic stem cells, we want to learn about them. And then we've talked a little bit about another kind of stem cell that's created in lab, that's a modified cell that's created in lab. So we'll go through those. And that's satisfying too, because we know something about stem cells and what they can do, and that also pulls it together. So we'll easily get through that on Thursday and start talking about chapter 20. Yep?
I have a question about.
Oh, I thought you were just going to sigh. I like that one. You just raise your hand. Just wanted to share that. Yep? Okay, sorry.
You've given us 2 different dates for changing in the term paper.
Oh.
One next week and one today, which is on the syllabus.
Oh, that's somehow a typo. Sorry. And Amal and Hannah, did you hand yours in because that following that date?
Yeah.
Yeah, that's, I don't know how that, I mean, maybe it's not a typo. Sorry. I've been thinking that it should be due like this, somewhat after our exam. You know, we should try to get our exam done. So if you are done with your term paper today and you can get it done like the weekend after our exam, that would be great. Sorry. Yeah, I created the syllabus and I don't remember, like I'm thinking, oh, that's a typo. But I guess at that time I had a reason for trying to get it done. But in previous years, the term paper has been due like after that exam, and that's fine with me. So, yeah. And Amal and Hannah, if you want to do anything more with yours, you can. Like if you were really scrambling, I apologize for that. I mean, it's great to get it done, right? And then move on to studying for the exam. But yes, let's say that if you're, you can finish it by the weekend after our exam, like that, get it done.
So I'm doing the second exam and our final.
Well, so you're not supposed to have any assignments due the last week of class. So we should get it done so we can, start moving toward the final. It's really dangerous to start saying, oh, I'll get my term paper done and, the day before the final. Yep.
Right.
I'm sorry about that. Thank you for that question. I didn't know. Yeah, and we're out of here, but yes.